hepatocyte, meaning it is what organ?
Liver.
Liver. The liver, if you ever don't know where something is happening, it is probably the liver. So, liver is going to have food sources such as glucose come in. Glucose goes through glycolysis, ends up making pyruvate, especially once you have your glycogen, and it'll do some glycogen and pentose phosphate pathway as well. Once you have as much glycogen as you need and you don't need the pentose phosphate pathway running, then you get even more going straight to pyruvate. Then pyruvate will go and become Acetyl CoA, which I am fond of drawing as just 2 circles because it is a 2-carbon molecule. Inside the mitochondria, squiggle, 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 we are going to have the TCA cycle, which we're familiar with. We're not going to do the whole TCA cycle today, but this is where we're going to kind of start. So Acetyl CoA. And do you remember the first reaction of the TCA cycle?
Is that a citrate?
Yes, beautiful. So we have Acetyl CoA plus oxaloacetate, or OAA for short, combining to make citrate using the enzyme citrate synthase. And so I'm just going to put arrow pointing to citrate, so that thing, synthase. Does citrate synthase use ATP?
No.
No. How do you know for sure? You can just say that you remember.
I remember.
All right, that works. A second way to know is that synthase never uses ATP. Synthetase uses ATP.
Okay.
So by the name of the enzyme, we know. Just a fun little thing there. All right, so these 2 guys come together to make citrate, and citrate is going to go ahead and be able to leave the mitochondria. So we're not doing the rest of the TCA cycle because we've gotten, at this point, we did glycolysis, we went through TCA for a while, and the liver got all the ATP it needed. And so now we don't need more glycogen, we don't need more ATP. What are we going to do with the excess? In the human body, we never just waste energy because that's a recipe for not surviving. And so we're going to do something with this energy. And what we're going to do with it is make fat. So this is where the fatty acid pathway begins. The first molecule that's like fatty acid pathway, I would say, is citrate. So citrate is going to be used so it can leave the cell. So we have synthase, citrate synthase on the inside. Then we have citrate translocase as the transporter, because acetyl CoA and oxaloacetate can't leave the mitochondria. There's no transporter for them.
Right.
But citrate there is. And there are reasons for this. Citrate, because it's a lot easier to regulate 1 or 2 molecules that can go through versus giving everybody a transporter, and then you have to regulate every different kind of transporter.
Right.
So,
well, there's so many processes that use acetyl CoA too.
Exactly. So, citrate synthase puts them together. Citrate translocase translocates it outside of the mitochondria, and then citrate lyase, Lyase. Citrate lyase will go ahead and break the citrate back apart into its original pieces. So we're going to have acetyl CoA and oxaloacetate. So oxaloacetate, what happens if mitochondria runs out of oxaloacetate? It's kind of a deep question. So if you take one piece, especially the oxaloacetate, out of the TCA cycle, then you aren't able to make any more citrate and you can't do the TCA cycle anymore. So running out of oxaloacetate is really basically saying you can't make ATP anymore, period, no matter what else you have coming in. And so we really want this oxaloacetate to get back inside. And it goes through a series of steps that we may have time to cover, or I may refer you to a slide on that one. But it goes through a series of steps to get back inside. So we recycle the oxaloacetate so that we don't break the mitochondria, essentially.
Okay.
Okay? The acetyl CoA is going to go ahead and be what we are going to use to make fat. So, with this, we're going to go ahead and have the enzyme it's either Malonyl CoA Carboxylase or Acetyl CoA Carboxylase. I'm going to write Acetyl CoA Carboxylase. I believe it can be called either Malonyl CoA decarboxylase or Acetyl CoA Carboxylase, depending on the textbook. I think Acetyl CoA Carboxylase is the more common.
Okay, so we need Biochem.
Yeah, awesome. Which is
B7.
B7. Yep. How did you know that?
Memorize it.
Memorized it. Awesome. All carboxylases. Beautiful. And if this is what Acetyl CoA looks like before, what do you think it's going to look like after it's acted on by a carboxylase?
It's going to add a carbon.
Exactly.
Where is it getting the carbons from?
That particular carbon, I'm not sure I know where that one comes from, to be honest.
Okay.
That's going to be a question for me down here. Carbon origin. CoA carb. Nice, thanks. Okay, and this is called Malonyl CoA. I always thought of the name Malone, like, you know, or not Sylvester Stallone, but that keeps coming to mind. And it's like Malone. It sounded like a gangster name or something. So Malonyl CoA. All right. So then after this, we're going to have a whole bunch of stuff that is not too complicated once you see what's actually going on, minus the fancy names. But there's going to be this is just a huge protein named fatty acid synthase.
Is this Malonyl CoA, isn't that what inhibits Cat1?
Yes, it is. Yep. And why do you think that might be?
Because you don't want to be making more if you need to break it down.
Exactly. Cat1 is part of breaking it down. This is part of making it. Doesn't make sense to be running both. All right. So before we talk about all the things that happen over here, and I'm going to erase all that, let's go ahead and do a review. So the way that I would review it is going to go ahead and say, okay, we make citrate out of acetyl CoA and oxaloacetate. Citrate synthase is the enzyme. Citrate translocase to take it out. Citrate lyase. So it's just synthase out lyase. Then we take the acetyl CoA, carboxylate it to malonyl CoA. So go ahead and see if you can find the picture and just take citrate stuff as group 1 and then outside the mitochondria stuff as group 2.
Okay.
And so there's just 2 groups.
Okay.
Good? Got it?
Yes. Yeah.
Beautiful. All right. I'm going to go ahead and erase this because this next one really helps to have multiple kind of pictures because the same enzyme, the FAS, fatty acid synthase, is going to do several different functions. So we're going to be watching it. It has 2 arms. Okay? So we have fatty acid synthase, and this is the fatty acid synthesis pathway. And it has 2 arms. Here's an arm, and here's an arm. This top arm, I'm going to make the top one the ACP site, which is like, great, lots of random letters. So I just call it the ACP catcher site, the ACP catcher. Catcher is what it does. Anytime a new molecule is introduced to this enzyme, it will always go to the ACP catcher first.
Okay.
It will catch it. So this is ACP catcher. And he'll catch things in order, as we'll see later. Now, down here we have a cysteine. Cysteine site, which I like to call the cysteine holder site. So we have a catcher and we have a holder, but I like to say ACP catcher. That way when I see ACP on the test, it's not like, is that the catcher or the holder? I know what they do, but I don't know what they're called. So cysteine holder. Okay, so in zoomed out terms, I think it helps to talk about the rest of what's going to happen in zoomed out terms first and then detailed terms later.
Okay.
So the fatty acid synthase is going to go ahead and grab an acetyl CoA, and then it will pass it down to the holder, and then it has its hand open. Then it will grab a malonyl, and it will always be malonyl from now on, and it will pass it down to the holder, chop off a little piece of it to make it sticky, and so one of the carbons comes off. And then grab another malonyl, stick it on, chop it off, and so it's grab and pass down and make sticky. Okay?
Where is this happening?
It's going to be this arm grabs them.
Right, but so we had
cytosol.
Okay, so it still is in the cytosol.
Yes.
Perfect.
So this is just a really big protein inside the liver cell.
Okay.
All right. So grab, pass, hold. There is one other step that I need to say. Once we have passed down the malonyl CoA has some hydroxyl groups, OH groups, sticking off the sides.
Okay.
If we left those there, this would be an unsaturated fatty acid, and it probably wouldn't even really be a fatty acid if there were too many of them, because you wouldn't have the bond strength that you need for, it wouldn't be a fatty acid. It would be something else.
Okay.
So each malonyl has 2 hydroxyl groups.
Okay.
So we grab a malonyl, we pass it down and just attach it like a Lego on top of the previous one, and then we're going to flick off the 2 hydroxyls by changing them into water, which we'll go into detail. You just add a hydrogen, basically.
Sure.
So grab, pass, flick off. Grab, pass, flick off. Repeat that over and over and over. Grab, pass, flick off until you've gotten a chain of carbons that is as long as you want it to be. And then that is what we call a fatty acid. So how does this do that? First, the ACP site, will grab an acetyl CoA, 2 carbons. Then it will pass that 2 carbons down to the holder site. And it does this using a very particular enzyme. Remember, this protein is frickin' huge. It has a whole bunch of little fiddly bits. This enzyme is actually part of the same protein. So if you get a question on the test that says malonyl acetyl transferase, MAT, M-A-T for short, malonyl acetyl CoA transferase, what is that? MAT, or maybe it says which of these enzymes is a part of the fatty acid synthase. MAT is part of the fatty acid synthase. So is the ACP site, so is the cysteine site. All of those are part of the same protein.
This is just a large protein.
Exactly.
Okay.
And so the MAT is going to be the one that allows, it takes it off the ACP site and passes it down to the cysteine holder site.
Okay.
Okay? So, pretty simple. Next, we have a free catcher site. So it's going to do something. And it's going to go ahead and grab a malonyl CoA. Okay? 3 carbons long. This one's malonyl CoA. This one back here was acetyl CoA. So I like to remember that it grabs the number 2. The number 2 comes before the number 3. Number 2, then number 3. So it goes acetyl CoA, then malonyl CoA. This still had the acetyl CoA down here. And then, I'm going to come around here for the next step. And we're going to have the MAT go ahead and pass down again. And we had, I'm actually going to change color right here just to make it really clear. We're going to make all of this green, because green is a happy color. All right. And so this malonyl CoA is going to be stuck on the end, and during this transfer, we are actually going to have this cut off. And so this will then float away as CO2. So the C in CO2 is this carbon.
Okay. Now the synthase is the only thing that is, we're not having any other molecules brought in to join those. It's just this.
Just the synthase is doing all of these actions. Everything we talk about.
Cutting, cleaving,
Yep.
attaching. Okay.
Yep. And there's a specific name for the cutting enzyme. It's keto something or another. It starts with keto, and I need to look at notes to know exactly what it was called. I don't recall that it was made into a question or made immensely important, but it was named on the second slide that you guys have. Second line of the second slide.
Okay.
Of that second slide of the block of paragraph of text and then all the green blobs underneath it. There's two of those. It's the second of those, not of the whole PowerPoint, excuse me. Okay. So it's keto something or another, like lyase or transferase or something. So,
so why, if it's going to cleave it anyway, why doesn't it keep just grabbing acetyl CoA?
So I'm going to guess, that's a great question. I kind of expected that you would ask it. Details.
Sorry.
No, it's good. This is actually why I enjoy tutoring. I think it's a regulation thing, because if the acetyl CoA, that's going to be present like all the time.
Right.
And if acetyl CoA is going to be a positive regulator of this whole process, it makes it harder to stop it when you don't want to do it. Malonyl CoA, you just turn off the one enzyme that makes malonyl CoA, whole process is shut down.
What is the rate limiting step in this process?
I believe that it is the malonyl. Ooh, wait. Nope. I think it's this. This is the rate limiting step. I do think the regulated step, which is different than the rate limiting step,
right.
Regulated step, I believe, is the Acetyl CoA Carboxylase.
Okay.
And so that one is the regulating step. This one, I believe, is the rate limiting step.
Okay.
So, and I'll double check that and email you rate limiting step and reg. Okay. Because I'm only 80% on those.
Sure.
So, yeah, so now we've got this, and I'm going to go ahead and draw in our little hydroxyls. Those are problems.
Yes.
All right, next. Got a circle, got catcher, got holder, got acetyl CoA, got the remains of malonyl CoA, and then we have little hydroxyls, OHs, coming off. Those OHs need to be handled, because otherwise it's not a fatty acid.
Right.
And so, how can we make an OH go away? This is probably a tough question, but anything come to mind?
Hydroxylase?
Hydroxylase, that would be a good enzyme name for one that cut it off. Another way to take care of it, that doesn't turn out to be the one that we use, is, yes, excellent, beautiful, dehydrogenase. So, we take in some hydrogen from NADPH, and the NADPH, and I think it's a hydratase rather than a D, but
okay.
this one I don't even think the name was in the slides, but it's part of the fatty acid synthase. But we take an H off of the NADPH and we add it right here.
Okay.
And then once it's like that, then we're able to use an enzyme, which I will double check and see if we have that name. I don't think we do, but just to double check, because this is like 6 months old memory now.
It starts from the innermost carbon, or it doesn't matter?
I don't know that that was ever covered, which one goes first. It eventually does it to both.
Okay.
So I do know that she won't test you on that.
Okay. It just for my own.
Right. So first, you stick a hydrogen on it, then you can bump it off because now it is H2O, which is really easy to get rid of. And then you just do the same thing again. So you do it with one first, and then do it with the other. Each time you're going to be using NADPH, and this H is what we're sending up there.
Okay.
So this goes there, and then it is one of these guys. So, yeah. And then repeat NADP, that's almost legible. H, right there. And we'll say it's that one. And then make it go away as H2O. And all of this leads to fatty acid synthase gets smaller every time I draw it. All right. Leads to a couple carbons, completely clean of hydroxyls. And so at this point, we just go ahead and I'm going to put a red asterisk here. Oh, I shouldn't do an asterisk. That means regulated step. I'll put a dot.
Okay.
This means repeat. Huh. Do you read music by any chance?
I do.
Okay. Let's see. Where would we go? Right here. There. So you repeat between those ones and just keep doing this segment over and over again. Grab a malonyl, pass it down, flick them off. Grab a malonyl, pass it down, flick them off. Flick off the hydroxyls.
Our PPP cycle has got to be like cranking it out.
Excellent observation. Yes. And that's why NADPH is important right there. It's because if you don't have NADPH, you can't make fat. And NADPH is also really, that pathway, I should say, is also for purines, because that's where the purines come out. So those are the two things for that pathway.
Okay.
Awesome, good observation. So we keep going until we get, I believe it's a 14 chain, yeah, it's a 14 chain fatty acid, and we call that palmitic acid. Palmitic acid. So we keep going until...
but can't we synthesize longer?
We can, yes. But this particular enzyme is...
just for...
Yeah, it makes the base fatty acid.
Okay.
And so it makes a 14-1. And then, once it has a 14 carbon chain, and whoop, there you go. There you go. 14 carbon chain, then it's going to go and be done. Removes. And that 14 carbon chain is going to do one of two things. It's either going to be used to make special fatty acids, Fa right there, special fatty acids. Those fatty acids are going to be a whole host of things that we don't get into in depth, but they will be done in the Golgi apparatus.
Okay.
So, Golgi.
Like arachidonic? Or casapent?
Yes, yes.
Okay.
So like arachidonic acid and anything that is either longer or unsaturated.
Okay.
And we can make them significantly longer. We do have limits, and we're really bad at making unsaturated. We can make some, but the reason we need so many in our diet is because we're crummy at making unsaturated fatty acids.
I don't know why it seems so easy.
Right? It's like just
bond it.
take a couple hydrogens away and it happens by itself. I mean, and hydrogens are wusses, so get your act together. All right. Beats me. It would be nice if we'd make omega-3s, but oh well. So we're going to make special fatty acids that serve some kind of purpose like that.
Okay.
Or we're going to go ahead and send them to be attached to backbone. So
a phospholipid?
A phospholipid is excellent, yeah. So phospho.
And where does that happen?
That happens in the cytosol.
Okay.
So phospholipid, phospholipids, and phospholipids can be a whole couple different families. There's glycerol backbones, there's sphingosine backbones, and all that kind of stuff. Yes. Most of the time, phospholipids, or I should say not just phospholipids, but also tags.
Okay.
Which is another backbone. It's still a glycerol backbone, but for a tag, you take away the phosphate. And so it's no longer a phospholipid, it's just a triacylglycerol.
Okay.
And that's why there's no phospho in that. So you're either going to make something on a backbone, or you're going to make some special single fatty acids. So, we've talked about the special single fatty acids in the Golgi. Phospholipids, cytosol, and then tags also cytosol. And there might, for the bulk. And phospholipids will be used however they need to be used. They're going to be sent wherever. Tags are going to be sent to adipose because they are just storage for all the extra energy that we've eaten with our Ben & Jerry's ice cream. And so we can store it as fat and pull it up later.
And once it's synthesized, it just accesses the hepatocyte and just gets delivered in the bloodstream to these adipocytes?
Yes, and it's delivered with a special lipoprotein.
Oh, so B100?
Yeah.
Okay.
Yep, B100 lipoproteins, aka VLDL, IDL, and LDL, which are all the same thing, with just as it gets less and less left in its Santa sac. So, yeah, and so that was the last thing I wanted to get to is VLDL, because that hooks it back into the digestion of fats, chylomicron story that we talked about another time, and that's going to be fatty acid synthesis. So, now, in order to make this stick, there was the first part, which had the citrate trio, and then the outside the mitochondria prep. Then there's this. So that was all like one thing with 2 bits underneath its heading. Then this is heading 2, which is going to be fatty acid synthase. And for that, I find it easiest, again, just go back to the catcher, the holder, catch, pass, hold, flick off. Catch, pass, hold, flick off. And then try and name, okay, catcher. What is the catcher? ACP site. What does it catch first? Acetyl CoA, and then malonyl.
Does it need NADPH to also flick off those hydrogens on acetyl CoA?
Acetyl CoA doesn't seem to have that. It only
you're right. It doesn't have the
has the hydroxyls on the malonyl CoA.
Okay.
So
totally.
That is unusual. I was thinking about that too, and it's like, did we not cover everything? Is there another little thing going on? Because there's probably another little thing going on, but we at least don't need to know. But it's good to know. So now I have a third curiosity. Acetyl CoA flick. Question mark, smiley face. Okay. So yeah. Go ahead and review catcher. So the catcher is going to be the ACP, and there's 2 things about catch you want to know. Name, what does it catch, in what order? ACP catches acetyl CoA first, then malonyl. Second thing, pass. Pass is going to be MAT. Malonyl, one of the things. Acetyl CoA, the other thing. Transferase. And so it's just named after the 2 things it can grab and pass.
So we're going to need 6 NADPH to make 14 carbons, right?
1, 2, yeah. 3, let's see. So 14 carbon long, so that's.
But we take 2 away for the acetyl
12,
CoA.
6, yeah, 6
Okay.
sounds right. Cool.
Okay.
Yep. And then what's the holder's name?
The holder is a cysteine?
Yes, the cysteine site. And there's a couple jumbled letters after cysteine sometimes, but it's the cysteine site.
Okay.
And then the cysteine, what is the second thing? There's 2 things at the catcher and there's 2 things at the holder. One's the name, then one's what they have going on there. So the holder has what going on? It holds it.
Flicks off.
Flicks off. It's the place where you're going to have stuff being flicked off to. And then that's the whole thing. So catcher, tell about it. Pass, tell about it. Holder, tell about it. Done. It's 14 and send off. And it's not bad at all.
No. how is this regulated in terms of, so we know we get it going, what's the regulation like stopping it?
I would imagine
So
that
it's
the
just
malonyl,
okay,
yeah,
it's just the
as
production.
soon as you don't have malonyl, you can't move forward. Oh, and incidentally, this is one of those semi-anal details that's like, we don't even know why this is important. We don't even know why the body does this, but fatty acid synthase is always a dimer.
Okay.
And so they don't help each other that we know. They probably do, but we don't know that they help each other. They just literally are linked together. And so this one is doing its own thing, this one's doing its own thing, but they are always twins. And so that could be a question. Which of the following is true about fatty acid synthase? Da 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 da. It's a dimer. Da 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 da. Dimer. Or which of the following is not true? So yeah. And then that's it. Any other questions? All right. Good deal. Catch you guys next time.